this year had just an unbelievable quality of applications and abstracts. We had a very difficult time selecting the top scoring science for this session, but we're going to kick off an amazing collection of talks today. These will be 8 minutes in duration. We'd like to ask the speakers to please stay to that timeline so we have the ability to have a Q&A session. The questions should be submitted through the Slido app, and then Dr. Gertner and myself will be able to read those out with the help of Dr. Alverdi. And so without further ado, we'd like to welcome our first speaker, Dr. Foster, who will be talking about cancer-associated fibroblasts and their heterogeneity. Thank you, Dr. Foster.
Good afternoon, everyone. Thank you so much to the Department of Surgery and the Holman Committee for the opportunity to present our research this afternoon. Today, I will be discussing a project exploring cancer-associated fibroblasts in desmoplastic solid tumors. So, solid tumors account for the vast majority of cancer deaths. The tumor microenvironment is composed of transformed cancer cells as well as non-transformed stromal cells, which include immune cells and endothelial cells, parasites, and cancer-associated fibroblasts, or CAFs. CAFs secrete the extracellular matrix components that make desmoplastic tumors, such as breast and pancreas cancers, firm. CAFs also interact extensively with other cells in the tumor microenvironment. However, CAFs have yet to become a treatment target in mainstream cancer therapy, which is likely in part because CAF heterogeneity and their roles in solid tumors has yet to be completely understood. Here we conducted a comprehensive investigation considering single-cell gene expression and chromatin accessibility in conjunction with spatial transcriptomics of CAFs across multiple solid tumor types and multiple species, with the aim of identifying commonalities among these fibroblasts that may represent promising therapeutic targets. To explore CAF heterogeneity in desmoplastic tumor types, we began by conducting single-cell droplet-based RNA-seq of syngeneic mice bearing an endogenous model of breast cancer. Modularity-optimized clustering of the resulting gene expression dataset identified 7 transcriptionally defined cell populations. Based on in silico fibroblast selection, we focused our analysis on CAFs, which were reclustered to delineate 6 populations of CAF subgroups. We examined these cells and identified characteristics and then provided names to each of these clusters. Cluster 2 CAFs we noted to exhibit an elevated expression of genes associated with mechanotransduction, including MGP, Gas6, and FOSB. And these were associated with focal adhesion kinase signaling. Clusters 3 and 4 also exhibited, or clusters 4 and 5 also exhibited related gene expression patterns. Looking at other transcriptionally defined groups, cluster 1 showed elevated expression of DPP4 or CD26, which is a serine protease which, along with its homologue FAP, is known to be upregulated in inflammatory conditions associated with scarring and fibrosis. The other 2 clusters also showed elevated expression of DPP4 and seemed to be associated with cells that might be modulating the immune microenvironment. Next, we applied CellChat, which is a recently developed tool to understand cell-cell interactions in a given niche using single-cell gene expression data. The aforementioned 2 master cluster patterns of CAF behavior were supported by this analysis. Pattern 1 highlights the mechanotransductive CAF subgroups, which appear to communicate primarily with each other in the extracellular matrix, whereas pattern 2 highlights these immunomodulatory subgroups of CAFs, which appear to primarily communicate with immune cells. Given our extensive analysis of mouse breast cancer CAFs, we next sought to evaluate how much these patterns of gene expression were recapitulated in human breast cancer. We examined cells isolated from human breast cancer patients. These single-cell samples were once again processed in the same way, and then we isolated our CAFs. Surprisingly, we found that there were also 6 subpopulations of CAFs identified in the human breast cancer. And similar to our mouse breast cancer CAFs, several of the human subgroups of CAFs expressed these mechanotransductive genes, including MGP and some of the other genes I mentioned, while the other 3 subpopulations appeared to have more of an immunomodulatory role. To evaluate whether the human breast cancer CAFs were analogous to the mouse breast cancer calves, we performed an integrated cross-species analysis using ortholog mapping in conjunction with a label transform approach. This allowed us essentially to project our mouse calf clusters onto the human calf embedding. And what we found was that there was a significant overlap between these. Taken together, these data strongly suggest a strong evolutionary conservation of calf response to epithelial breast cancers in the form of these 6 transcriptionally delineated clusters. Next, we sought to evaluate whether the local fibroblast response to epithelial neoplasia was conserved across multiple tumor types. In order to do this, we examined pancreatic ductal adenocarcinoma, or PDAC. Similar to breast cancer, PDAC is associated with a strong desmoplastic response, and calves are among the most common cells in PDAC. We examined resected tumors from PDAC patients, and we identified among the calves from single-cell sequencing, we found 7 transcriptionally delineated groups. Next, we used a similar label transform approach to overlay our human breast cancer calves and our PDAC calves to identify 6 transcriptionally defined shared clusters between breast and pancreas. And remarkably, 2 of these clusters seem to delineate primarily immunomodulatory clusters, whereas 3 of them were more mechanotransduction gene expression, while one of the clusters was only represented by the PDAC patients, and this was primarily associated with EMT genes. So these results support this evolutionary conservation of the fibroblast response to epithelial tumors from multiple organisms to form transcriptionally defined clusters. To evaluate the epigenomic changes associated with fibroblast activation in the context of solid tumors, we conducted a series of single-cell ATAC-seq experiments in parallel with our RNA sequencing experiments using our endogenous mouse tumors. We identified considerable heterogeneity in the accessibility profiles among the calves, which were clustered into 4 epigenomically designed subgroups. Then we performed a cross-platform analysis to link the mouse breast calf single-cell ATAC with our earlier RNA data. This resulted in 4 shared mapped clusters. And when we examined the epigenomic landscapes of these 4 clusters, we found that 2 of them mapped significantly to the mechanotransduction genes and showed increased accessibility among some of the aforementioned genes, such as MGP, whereas the 2 other clusters showed significant accessibility around DPP4. Next, we wish to further explore the significance of calf heterogeneity in solid tumor biology. As such, we applied the Visium platform to analyze gene expression while retaining spatial information. And we conducted spatial transcriptomic analysis of whole tumor sections taken from our endogenous mouse model. Using the transcriptional profiles identified on our single-cell sequencing analysis, we were able to predict all the spatial contributions of our 6 calf subtypes amongst the tumors. And we found that these largely followed the patterns we would expect. So we found that the immunomodulatory subtypes seemed to map to areas that had a high proportion of immune cells, whereas the mechanotransductive subtypes seemed to be more in the center of the tumor and mapped more towards areas that were sparse in immune cells. Finally, in order to validate the relevance of our data, we did several functional experiments. The first, we knocked out FAK in mice, and we isolated the calves from these, and we found that when FAK was knocked out, we saw it at a protein level that the genes that we found to be associated with these showed decreased expression. We also found that in the FAK knockout mice, the tumors had a significant change in size, suggesting that actually the mechanotransduction subgroup of calves support tumor restriction and growth. The next thing we did was we identified a public data set of, or a published data set of immunomodulatory data, and we found that the immunomodulatory calves seem to respond primarily when immunotherapy is applied to a tumor. This just summarizes those data. So in summary, while current therapies focus largely on transformed cells, we believe that CAFs represent an understudied therapeutic target that could improve multimodal chemotherapeutics. We conducted a comprehensive investigation considering single-cell gene expression and chromatin accessibility in conjunction with spatial transcriptomics of CAFs across multiple solid tumor types and species, and identified commonalities across these fibroblasts that really may help identify shared components of these cells that could improve therapeutics. We found that CAF heterogeneity is conserved amongst both solid tumor types and species, and really identified these superpopulations, the mechanotransductive and immunomodulatory. These categories are governed by specific changes in chromatin accessibility and are spatially distinct. So primarily, I'd just like to acknowledge Dr. Longacre and Dr. Norton for their extraordinary mentorship and the opportunity to do this research. I'd also like to acknowledge Drs. Wapner, Gertner, Wan, and Howard Chang in the Department of Dermatology, who are co-PIs on this project. Next, I'd like to acknowledge Michael Januszyk, who's also a presenter today and who's a co-first author on this paper, as well as other co-authors and members of the laboratories, and our facility funding and academic support.
Well, thank you, Dr. Foster. That was an amazing body of work. I'll remind the audience that you can now type in your questions to Slido and we can read them out. And while we're waiting for those, I'll kick it off. I'm wondering where these CAFs come from and how they get there. Do you think they have a common origin and then branch out? Do you think they have clonal expansion? Or do you think they all arise in parallel? And the reason I'm asking this is, can you shift them from one way to another for therapeutic purposes?
Certainly, that's a terrific question, and certainly is a hotly debated topic in the literature in terms of how many of them particularly are locally derived or of systemic origin. In our data, which I didn't have a chance to show today, but we do have quite a bit of data supporting the idea that the vast majority of are locally activated and really do proliferate clonally in response to neoplasia. Which is promising in that we really think that this is probably a local response and potentially why it's shared across different organs and different species. And I think it provides promise for the possibility of really being able to therapeutically target them, because if it is a local response, then we should be able to really delineate specific therapies that are going to primarily have effect locally.
Thank you, Dr. Foster. That was spectacular, elegant work. I was getting exhausted just watching you run through all the data. And thinking,
I don't have very much air to spare right now, so I'm sorry if I was panting.
No, not that. I meant just all the body of work. The body of work was just so expansive. It was a ton of work. Maybe my question is along the line of yours, which is that, is there a secretome of the primary tumor that provides an environmental cue that drives these cell lines toward one phenotype versus the other? And how, can you talk about the limitations of this? Because the work is, while it's amazing, it's largely descriptive because you haven't sort of knocked down what is the driving signal from the tumor microenvironment that actually, clonally expands, one phenotype versus the other. And how might you resolve that question? And can you talk a little bit about the limitations of what you can and can't do in your study? Enjoyed it very much. Congratulations.
Thank you so much for the question. I really appreciate it. Yeah, certainly this is largely descriptive work, which I think is kind of how one has to start a project to ask this sort of question, because we do need to validate that our hypotheses are relevant in that regard. I have started, and I kind of just mentioned them briefly, some knockdown studies, at least to preliminarily kind of try to functionally validate these, and it's been exciting to, thus far, things to seem to line up pretty well, which is exciting in that regard. And I think really being able to trace what happens to tumors when we modulate these populations is really the first step in that. And then, of course, to follow that, really looking at it from a protein level, and are there things that we can actually target once the tumor has developed, which is definitely the exciting next step. And I really look forward to doing those proteomic analyses once kind of I've validated things a little bit more at the gene expression level.
One quick question from the audience: is engrailed 1 expressed in these fibroblasts?
The million-dollar question. Shamik's around here somewhere. It is, although not as highly as we would expect. It seems like engrailed is something that is primarily a skin associated fibroblast marker, and we do see it at lower levels in the breast, but not very much at all in the pancreas. So it seems like it's there's some variability there, and that heterogeneity is really interesting to think about. So. Thank you so much, Dr.
Move on to the
Thank you so much.
Thank you. So wound healing is a complex physiologic process that represents a model system in which to study tissue repair more generally. Fibroblasts represent the key mediator of wound healing, responsible for collagen secretion, ECM production, and coordination of the inflammatory response. However, research in wound healing is limited in part because fibroblasts themselves represent a heterogeneous cell population, making traditional tissue analysis techniques more difficult. Our lab has recently demonstrated that in the context of mirroring wound healing, fibroblasts are locally derived, are activated in response to local environmental cues, including mechanical signaling. Using the rainbow confetti mouse construct, we've shown that these cells proliferate radially inward in a polyclonal fashion to fill the wound gap. And in this process, going from the outer edge to the inner edge of the wound, their transcriptional programs shift considerably. In the last year, Deshka Foster and I have spent considerable time working with the folks at 10x Genomics, as well as Stanford's functional genomics facility, to apply next-gen omics technology to study fibroblasts in the context of wound healing. I'm first going to talk about single-cell RNA-seq, which we applied to mouse dorsal excisional wounds. We looked at outer and inner wound regions separately using tissue microdissection, and we found 4 transcriptionally defined subpopulations, which you can see in the top middle panel here. 4 clusters, one of which in green had a particularly less differentiated transcriptional program. It was also preferentially expressed by outer region wound fibroblasts. So before I go into more detail, I want to quickly mention that we also did parallel analyses using single-cell ATAC-seq on these same phenotype paired mouse tissue samples. So for those not familiar, ATAC-seq refers to the assay for transposase accessibility of chromatin. Essentially, it looks at DNA open versus closed reading regions developed by our co-author Howard Chang here at Stanford. This lets us look at more upstream processes. So instead of looking at transcriptional counts, we're looking at the DNA itself and whether or not it is open or closed, and we expect this to precede the eventual transcriptional processes. I'm going to skip through this slide and mention that when we look at fibroblasts in the context of chromatin, in wound healing, we see 6 populations defined based on ATAC accessibility. Working with the folks in Will Greenleaf's lab, we managed to integrate these 2 data sets together using single-cell RNA-seq and single-cell ATAC through the novel platform Archer. And this lets us construct multimodal data constructs that incorporate both single-cell RNA and single-cell ATAC information. This gives us an unprecedented lens in which to study the behavior of these cells during the wound repair process. And the net, it's difficult to interpret these slides over this time course, but we see 4 populations again, one of which we've named all 4 again, as Deshka did for her CAF work. And one persistent phenotype that we observed is what we've called the mechanofibril fibroblasts, which are in green here. And they have elevated expression and accessibility of key fibrotic markers, including PDGFR-alpha and focal adhesion kinase. Among other markers, going back to the last question, we did see that engrailed was expressed by these cells. Engrailed is a favorite marker of the Longecker and Gertner laboratories. We published on it in Science 5 years ago, and then Shamik published it again, a more updated version last week in Science as well. So it was consistent with these observations. So lastly, we examined, given that we saw differences between outer and inner wound fibroblasts at single cell and gross levels, we wanted to apply spatial transcriptomics to this study as well. Unlike in tumors, which are in some sense amorphous masses, wounds have directionality, and this helps us to really use more spatial information in this context. So what I'm showing now are 4 distinct time points in the wound healing process: unwounded, post-op day 0, and then post-operative day 2, 7, and 14, which are the 4 columns on the right. Visium, or spatial transcriptomics, essentially takes tissue histology slides that are flash frozen in OCT medium, and then essentially it plops them into a micromechanical plate, and you get what we can refer to as spots. One spot corresponds to 1 to 10 cells worth of mRNA. So the tissue section goes into these plates, and then it gets sequenced as you would any other high throughput nucleotide material. So doing that, and this is the last slide I want to talk about in detail, we can evaluate the phases of wound healing with temporal and spatial resolution. So what you're seeing here on the left, again, we have 4 rows corresponding to 4 distinct time points in wound healing. And then I have 3 columns, each corresponding to 1 gene. So when you apply Visium, you get essentially a tissue histologic section broken up into these spots. And for each spot, you get the entire 20,000 dimensional transcriptome. So you're seeing 3 columns here of the candidate 20,000 that we could show. So the leftmost one shows KRTB6, a keratinocyte marker. You can see in unwounded skin, it is present at the epidermis. And after the wound, it's not there, obviously, in the wound center at day 2. By day 7, they've migrated toward the wound center. And at day 14, in a scar, we have a fully epithelialized tissue section. PDGFRA is a good marker for fibroblasts. We can see that they are largely dormant at unwounded and post-op day 0. And then at post-op day 7, they migrate toward the wound center and are most active at day 14. Finally, you can look at MSR1, a macrophage marker, again, dormant at days unwounded and day 2. And then at day 7, we see this proud flesh area near the wound center where we have a bit of inflammatory milieu. And then by post-op day 14, these cells are still producing this in a sustained fashion. So furthermore, we can use this technology to impute the spatial location of our previously defined populations indirectly to track them in both time and space in these sections. And I will just mention briefly that this allows us to demonstrate that fibroblasts differentiate as they migrate toward the wound center. As they do so, they have programs that become less mechanoresponsive and more fibrotic. These changes are preceded transcriptionally by epigenomic changes, and we can also do some informatics gymnastics to impute the spatial epigenomic properties of the resulting tissue sections, which I won't get into here in detail. And then if we do block mechanical signaling, we see that this well-coordinated process is disrupted, and we end up with less clonal, more disorganized scarring as a result. Sorry to rush through this very quickly. This work was just uploaded to bioRxiv. You can find it in full there. If you Google BIORXIV space my last name, you can download it and take a look. Happy to answer questions now or by email. Thank you.
So I encourage everyone to submit your questions via Slido. While that's going on, or I'll ask a question. So the fibroblasts are changing as they go from the outside to the inside of the wound. But is that because they're going from the outside to the inside of the wound, or is that because of the passage of time? How do you separate, is it time dependent or is it spatially dependent? And if it's spatially dependent, how do the fibroblasts know where they are?
Right, that's a good question. And we can determine that by looking at sections at different time points. When we get a post-operative day 7 section, we have fibroblasts that are, let's just say it's point X on the periphery. We can look at that same location at days 2, 7, and 14. And we do see that they change irrespective of migration, just by passage of time, they do change. But they also change through movement in space as well. So it's difficult to deconvolve the 2 factors there. And we only have, I think, 8 sections in this study. So we probably need another order of magnitude worth of data to really pin down that question.
Not yet, okay. So I'll ask a follow-up. Oh, yes, please. Yeah, please.
Congratulations. Very nicely done, beautifully displayed, and exciting work. Because you can't, sequence your way to truth. I mean, we all know that, and I don't know this field very well, but it strikes me that during mechanical-transduction, there are going to be small molecules beyond proteins that form, because you use the term environmental cues. It's those stromal environmental cues that are going to direct the phenotype. Can you talk about how you might measure things other than proteins that might be secreted that are playing a role here, that's the sort of piece in the interactome that you're missing? And how might you resolve that? Thank you.
Yeah, thank you. That's a really good question. And I think the short answer is because we can't sequence the interactome per se, it's hard to get those assays scalable to the level we need. So we have to get there indirectly instead and kind of predict what might be present in the environment. And you can do that using a lot of different modeling systems, including the cell chat that Deshka mentioned. But you're essentially just taking good guesses and then testing your guesses sequentially in hopefully a data-driven fashion. But yeah, unfortunately, we don't have the capabilities to. That would be great to do, single cell interact, or single, I don't know what the resolution would be, single spot interactome mapping for these tissue. Someday.
So final question. So when human wound healing, the fibroblasts don't come from the edges, right? I mean, they come from like the bottom. When the keratinocytes come from the edges, but you see granulation tissue, which is composed of endothelial cells and fibroblasts. How did you eliminate that from your mouse model?
Right. Yeah. So that's definitely true. I think traditionally in wound healing we use the Galliano model from your lab at NYU, and that are some variant thereof. But in this context, we essentially dissect the initial punch biopsy to the level of fascia and remove everything above it. So we're kind of it's not truly physiologic in that sense, that we don't have the inherent granulation over underlying bone, muscle, or fascia. So that's the limitation of the system, to be certain.
All right, I think in the interest of time, we're going to have to move on. Thank you very much, Dr. Januszyk.
Thank you so much for that introduction, and I just want to take a moment to thank you for allowing me to present my research today. So osteoarthritis is a degenerative joint disease affecting 15% of the adult population. And because of various factors, your body is unable to regenerate articular cartilage. And this can lead to a debilitating loss of function in the affected areas. Multiple factors lead to the progression of this disease, including age, obesity, joint trauma, and inflammation. And so the goal of our project is to regenerate this articular cartilage. And this is not a trivial thing to do because your body does not, as an adult, spontaneously regenerate articular cartilage. So multiple therapies exist that attempt to treat this disorder. So one option is medicine. You can also have a joint replacement or microfracture surgery. And this involves the drilling of small holes into the diseased site, and this allows bone marrow to seep into that joint space. And while this can produce a type of tissue that can alleviate symptoms for a period of time, eventually this tissue degrades, and so it's not a long-term solution. Now, in our lab, we have identified the mouse skeletal stem cell, or SSC, and its downstream lineage. So this SSC gives rise to the BCSP, or the bone, cartilage, and stromal progenitor cell, and this in turn gives rise to cartilage, bone, and stroma. And in addition to this, we have also identified the human SSC and its downstream lineage. Furthermore, we've identified that we can utilize specific factors to skew the fate of the SSC. So if we cut off the lineage to the bone and stroma, we can actually skew the SSC towards producing cartilage. Now, we wanted to start by looking at the cell populations and the tissue construct at the site of the articular surface. When we looked at the clonality of the cells at this articular surface, we found that there was a decrease in clones with an increase in age. And when we looked at the tissue composition, we found that there was drastic changes throughout the lifespan of the mouse. So this is a pentachrome stain where we have cartilage staining blue and bone staining yellow. And you can see that there is a decrease in that cartilage at the articular joint. And looking at the cell populations, we found that there was a decrease in SSC and BCSP numbers with age. And then we plated these cells for colony-forming units to see how their self-renewal changed with age. And we can see that there is a decrease in CFUs in the adult mice compared to the P3 pups. So then we wondered how focal microfracture affects the SSCs at the articular surface. And here we have the microfracture site outlined in the white line, and you can see that there is an increase in clones present in that microfracture site when compared to the surrounding tissue. And then when we look at the pentachrome stain here, we have an increase in cartilage compared to the surrounding tissue. And then we wanted to stain this to look at the cell surface markers of the tissue, of the cells present in this tissue. And we found positive staining for Col1, Col10, and MMP13. And these are all fibrocartilage cell surface markers. And we found minimal staining for Col2, which is an articular cartilage cell surface marker. And this is interesting because at the articular surface, you really want to see articular cartilage. It's a strong, stable cartilage. But what we have in response to this microfracture surgery is a fibrocartilage. And we saw, as a response to the microfracture surgery, we have an increase in SSCs and an increase in cell division of these cells. And we see this at weeks 1 and 2 post-surgery. However, we have a drop-off of this response by week 4. So this is a very transient temporal response. And we wondered whether this was a local SSC response or it was circulating SSCs being called to the site. So we used a parabiosis model and we performed microfracture surgery on the non-GFP mouse. And we found that there was no GFP positive SSCs or BCSPs at this site. So we gathered from this that it was a local response rather than circulating cells. So then we wondered if this microfracture surgery was causing intrinsic changes to SSCs. So we used transplantation surgeries to identify this. When we performed transplantation in the renal capsule, we found that the adult non-microfracture control produced a high amount of bone in the graft. However, the microfracture SSCs produced various different tissues, including cartilage. So then we looked at transplants on the articular surface, and we again found that the adult control was producing a high amount of bone, whereas the microfracture cells seemed to be producing more of a fibrocartilage tissue. And when we stained these for cell surface markers, we found an increase in Col1, Col10, and MMP13 stains, and minimal Col2. We also did gene expression analysis, and we found that cartilage and cell cycle-related genes were increased in response to microfracture surgery, but inflammation-related genes were actually decreased. So we wondered whether we could add additional factors at the site to skew the production of the tissue. And so here we utilize a hydrogel with PBS, and we see minimal regeneration at the site. So we added BMP2 to our hydrogels, and we found that we could increase the regeneration of tissue. However, it was more of a bone and fibrocartilage tissue that we saw. So we decided to add BMP2 with soluble VEGF receptor, which is a VEGF inhibitor, and we were able to produce cartilage. And while this was a really exciting and interesting result, we wanted to ensure that this was articular cartilage that we produced. So we stained for our markers again, and we found positive staining for aggrecan and Col2, which are both articular cartilage markers, and there was minimal staining for Col1, Col10, and MMP13. And we saw this result 16 weeks out, so we were able to verify that this was a stable articular cartilage and not some transient tissue. Then we wanted to identify whether we could see the same result in a human setting. So we utilized fetal phalanges and transplanted them in the dorsums of mice and performed microfracture surgery with transplantation of our factors. And again, we saw with our BMP2 and soluble VEGF cohort, there was an increase in cartilage production at the microfracture site. And we identified that this was a result of the human cells producing this tissue. There were no invading mouse cells. So in summary, we found that maturity leads to a reduction in articular SSCs, but focal microfracture leads to amplification of the SSCs at this articular site. And this is a local response, not due to circulating SSCs. We also found that focal microfracture leads to cell-intrinsic changes in SSCs, which we identified using our transplantation assays, and we also saw that we could skew the tissue production by addition of different factors. So our addition of BMP2 and soluble VEGF receptor could promote the production of articular cartilage. So I just want to thank everyone in the Charles Chan lab for all of their help with this project, as well as Matthew Murphy, who actually started this project. And I also want to thank our collaborators in the Longacre, Wiseman, and Fanyang labs. Thank you so much for listening, and I'm happy to take any questions.
Congratulations, that was a beautiful presentation. Having just been through this surgery myself, I learned quite a lot. So I guess I'm wondering why blockade of VEGF promotes the cartilage growth. I would have assumed that, taking away blood flow would reduce the ability of these cells to regenerate. So do you have any thoughts about that?
Yeah, so what's interesting is BMP2 will actually promote the self-renewal and differentiation of our SSCs. But if you allow it to just go forward with its natural fate, the BMP2 will cause it to produce bone. So the VEGF, which is a highly vascularized tissue, whereas cartilage is avascular. So if we block off vascularization using the VEGF, we actually cause these cells to produce cartilage instead of go towards that bone fate. I do think we could potentially create a better therapy by making it temporally, adding these factors temporally. So I think adding BMP2 first, followed by VEGF, so you get a little bit of that vascularization, which I think is important for the production of this tissue, but you do need to cut off that vascularization at some point so that these cells will go down that cartilage fate. Yeah.
Fantastic job. Thank you very much. I learned a lot. And every time, you went through your presentation, I thought, hey, what about BMPs? And then you presented, and I said, hey, what about degradation pathways? And then you presented some MMP13. I said, what about blood supply? And then you threw in the VEGF. And so it kind of stumped me each way. But I do want to bring up this point of synthesis versus breakdown, because collagen obviously remodels as a function of a balance of synthesis and breakdown. And so I wonder if the durability of your model would change if you focused a little bit more on those proteases that actually break down the tissue over the long haul. And how are you going to consider that in your next set of experiments? Thank you.
Yeah, thank you so much for that question. I think that's really important. It's not something that I have explored so far, but I think that's a really great point. And, we have looked at the tissue composition using atomic force microscopy, and we found that the mechanics of the tissue is very similar to the uninjured cartilage. But I think moving forward, that's something that we should definitely keep in mind and kind of come up with experiments that can address that.
Okay, and a couple of quick questions from the audience, and I would encourage people to keep submitting these. First, how long after injury can you still skew these skeletal stem cells? And secondly, are you surprised that the microfracture reduced inflammation in this model?
Yeah, so first, we do see that these SSCs have a restricted time point in how they respond. So we do see an increase in SSC proliferation in response to surgery at 2 weeks post. So I think we do have to add these factors close to the microfracture surgery. And I think ideally it's at the same time. And then as to the second part of that question, I do find that kind of surprising, and I'm not actually sure why it reduces all of those inflammation-related genes. I think that's a great question, and I would love to explore that further.
Great answer. Congratulations on a beautiful project.
Thank you so much.
Hello, everyone. I'd like to thank the program committee for this opportunity to present our work. I have no disclosures. With 21 degrees of freedom in the hand and 6 at the wrist, the upper extremity is capable of numerous sophisticated movements. Its loss is devastating, and over 500,000 Americans are living with upper limb loss. Active prostheses are either body-powered, controlled by the contralateral limb using cables, or are externally powered, either electric or myoelectric, controlled through EMG signals. The problem with these solutions is an inability to replicate the degrees of freedom of a normal hand and the lack of a sensory feedback loop. Next-generation prosthetic systems provide increased range of motion and dexterity. Some are capable of 5-finger movement and 20 degrees of freedom. The current limitation is the interface between the machine and our body's neural circuitry. Cuff electrodes lack resolution and selectivity, while invasive electrodes, such as the Utah slant array, are fragile, traumatic, and result in a progressive foreign body reaction with signal degradation over time. The TIME, or Transverse Intravesicular Multi-Electrode Array, is a recent attempt at creating a flexible intraneural device, but it's still relatively large with a limited channel count and resolution, and it also induces a foreign body reaction that results in loss of signal over time. Targeted muscle reinnervation and regenerative peripheral nerve interfaces are the cutting edge of neural interfacing, but they are still EMG signals with limited resolution, they lack a sensory component, and they're subject to foreign body reaction to the electrode. The ideal solution would be an intraneural electrode with high resolution and fidelity, allowing for single neuron selectivity and two-way communication, combined with mitigation of foreign body reaction. Foreign body reaction has been compared to an ongoing wound healing response that remains unresolved due to the presence of foreign material that cannot be phagocytosed, and that leads to degradation and encapsulation of the implanted electrode, as well as damage to the surrounding tissue. Increasingly, the role of mechanical forces, as you've heard in the presentations before this one, and more specifically, a structural and mechanical mismatch between the foreign body and the native tissue has been implicated in the process. So why not make the implanted biomaterial as much like the target tissue as possible? Da Vinci designed the hull of a boat based on the line of a fish, while his ornithopter design was inspired by birds. And recently, Charles Lieber's group in Harvard created a mesh-like neural probe that mimics the structural and mechanical properties of neurons in the brain. They showed that it elicits a minimal immune response, prevents gliosis, and has stable single-unit recording at 3 months out. Unfortunately, 20% of mice sustained intracerebral hemorrhage and critical cerebral edema during insertion. So we opted to apply this biomimetic approach to a peripheral nerve electrode. And with our collaborators in material science, we nanofabricated electrodes that are 10 micrometers wide, mimicking the size and mechanical properties of native axons. Through an iterative process, we designed and fabricated what we call the NeuroStitch device, which can be inserted with a pull-through methodology. It is 150 times more compliant than the TIME electrode I mentioned previously. Here we see a 10-0 suture tied to the device in the first panel and the needle glued directly to the device in the second panel. The sciatic nerve of a mouse is exposed in the third panel. Here we see fluorescent imaging of the device inserted in a Thigh-1 YFP mice, which demonstrates the structural similarities of our device and the native axons. We wish to assess the acute nerve injury caused by insertion of our device, and using the sciatic functional index, we demonstrated that insertion neuropraxia resolves at 1 week and that function remains normal at 6 months post-implantation. Of course, we also need to make sure the electrode actually works. Here's our setup to assess its function. Selective stimulation of individual channels representing a single electrode string resulted in contraction of a specific muscle group in the mice hound limb. Here we see the thigh musculature being stimulated. Here we see the fourth toe being stimulated. And here we see the fifth toe being stimulated. We can record from single neurons with microsecond time resolution on 32 channels. On the left is the electrical reading as a waterfall plot, and on the right is the electrical reading on all the channels when a specific neuron fires. We did some preliminary analysis of the fibrotic response to our device, and at 6 months found that it was comparable to sham, suggesting a minimal foreign body reaction and the potential for long-term stability. To allow for chronic recording and simulation, we designed a mouse backpack for housing the hardware. Of course, the best laid plans of men are often ruined by mice, and it required a few iterations before we developed a housing for the circuit that they were unable to open and could be secured to the mouse. And we were able to successfully implement it. In conclusion, we've established a proof of concept for a biomimetic, ultra-small and compliant peripheral nerve interface in a mouse model. Our NeuroStitch causes minimal and self-limiting insertion injury, can selectively stimulate and record, and does not appear to cause a fibrotic response. Our goal is to one day provide patients who've lost a limb with a neural prosthetic capable of performing natural motion with sensory feedback. And our future steps include assessing signal stability over time, expanding our analysis of the foreign body reaction, and scaling up our channel count. We're also working on disrupting the transmission of action potentials to address chronic pain. I'd like to thank my mentor, Dr. Gertner, and our collaborators from the Malosh Lab. Thank you.
Thank you, Zeshaan. Obviously awkward for me to be the one asking the questions, but what are the computational requirements if you have all these channels, you have all these stimulators, and you're going to have these closed loop systems? I mean, are the computational, does that kind of a computational feedback loop exist, or is that beyond current computational capacity?
I think that's an active area of research. There's a Stanford Neural Interface Center here that works primarily on brain-machine interfacing, and they're working in that area. I don't personally have that expertise, but our collaborator in the Malosh Lab, Eric Zhao, as part of his work, is developing some machine learning algorithms to help with that.
Great.
So it certainly is in development. Yeah.
Outstanding work.
Thank
I
you.
got to say, you're kind of scaring me. This is some kind of doomsday machine thing happening here.
Well, did you recognize the hand?
Yeah, it was very, very
Luke
scary.
Skywalker.
But it's actually just such elegant work, and I congratulate you. I just wondered, as you was putting it together, a couple of things. Maybe it's the same question you're asking about how you compute how the harmonic is running through the artificial nerve. It's not just amplitude and length and all that other stuff. It's more of a dynamic, it's some kind of equation of motion or something. How do you account for that? The second is, I was thinking about most nerves have these, internuncial spaces because they need to be dampened and turned on and turned off at different times. Can you make other nerves interact with that single nerve? Is that the hope? Or because you can't just have one nerve going from the cortex all the way down to the end effector finger muscle, right? It's going to, for coordination to happen, it's going to have all that.
Yeah. So great questions. And to address the first half of the question, which you said ties in with what Dr. Gertner was asking as well, a lot of this work is actually being done to some extent on that side of things by labs like Paul Sederna's lab at Michigan, because though their interface technology hasn't moved forward as much, the post-processing work is being done. And a lot of it is actually simpler than you think, in the sense, if what you're asking me is, you can ask the patient to perform a certain action, read the signal, and then see which of your channels lights up. And so that's one way of doing it. With regards to the wavelengths, the amplitudes, we do a lot of EMG nerve conduction studies for patients with nerve compression syndromes. And so some of that data has been collected over the years from that purpose as well. And then the second half of your question was talking about coordinating different nerves. So each nerve, like once you're at the postganglion, that single nerve does run all the way down to the muscle it targets, but each nerve obviously has multiple axons. It carries fibers that cross over synapse. But we actually have a pretty good understanding of, in the upper limb specifically, which nerves target which areas. A lot of our surgical interventions, nerve transfers rely on that knowledge. So I think that's not as much of an issue, but this other benefit of the recording side of this technology is we could actually confirm at a functional versus recording level whether what we understand about like the maps of nerves is true just by recording over time, just collecting that data over time. Yeah.
So we have a couple of questions from Slido. One is, how, as you get into experiments, how do you promote reproducibility with the trajectory of each stitch being somewhat different, and then how do you you're doing an N of 1 experiment over and over again?
Yeah, I think that's a great question. I think that certainly at this stage we're very much in the design proof of concept part of it, and eventually we would want to get into a more reproducible insertion and recording methodology. Something we've thought about, and recently we've actually gone to a microwire insertion approach as opposed to a suture needle. We started with the suture needle because we're microsurgeons, that's what we do, it's what we knew. But with these microwires, we can actually scale down to like a 30 micron wire, and then that can be multiplexed. It could be put onto some sort of backing that allows a press fit insertion. And so certainly there are possibilities for standardization.
And then the last question is really theoretically compared to like the RPNIs and those sorts of things. Theoretically, how many degrees of freedom could you get with this kind of approach with all the different channels and in terms of, I know you're not there yet, but do you think it's possible to reproduce all the different degrees of freedom in the hand?
I think that if we can scale our channel count, our number of electrodes, then I think absolutely. I think in terms of the degrees of freedom in the hand, crudely, we could probably do it with what we have right now. I think that's certainly possible. I think that when you're talking about like regulation of force amplitude, picking up a grape versus grabbing a soda can versus, grabbing onto a ball tightly, that type of work would require both scaling up our channel count and I think a better understanding of wave amplitudes and that post-processing of the information that we're getting.
Oh, great. I think we're going to have to move on, but thank you very much, Zeshaan.
Thank you for the introduction and the opportunity to speak here today. I want to start off by talking a bit about the traditional view of wound healing, and then a bit about what we will be talking about today. In the traditional view, you have an injury, you have inflammation in that local area, you have the proliferation of fibroblasts that are going to be laying collagen, and then you have scar formation and eventual remodeling. There's been a lot of recent literature about this subcutaneous layer and how it might be contributing to the wound healing process. Some literature has shown that there's a transdifferentiation process between fibroblasts and adipocytes. Our group is really interested in scarring and wound healing, as you've learned today. And so we wanted to look at this adipocyte to fibroblast transition and how those fibroblasts might go on to impact wound healing. These are some of the literature I was referring to. Some of the goals we had for this project, we wanted to determine the roles, if any, of adipocytes in wound healing. Of course, we first have to confirm that they are participating, and then we wanted to characterize the ways in which they were participating and talk a bit about when in the wound healing process they were becoming involved. Finally, we wanted to look at what cues might be important to getting these adipocytes involved in wound healing. How do they know to transition? The first thing we did was we took a RFP mouse, a mouse that expressed red fluorescent protein in all of its cells, and we took out the fat cells and we injected those into a wild-type mouse. After a couple days, we injured an area around that injection site. As you can see, in the unwounded condition here, in that first 2 rows, we don't have really any engraftment of these cells. So there's not a lot of space for these cells to live, and so they probably just die off. However, in the postoperative day 7 and 14 conditions, we do see some engraftment in this red column. And we also have adiponectin expression. So adiponectin is a mature adipocyte marker that co-expresses with this RFP. We expect that because we're injecting fat cells. But we also see COL1, which is a fibroblast marker, co-expressed with some of these cells. And so what we think we're seeing here is adipocytes are surviving in the wounds, and some of them are transitioning to fibroblasts. Now, there are some challenges with this model. It is possible that while harvesting this fat, that we got even just a single fibroblast, and injecting that fibroblast into the wound or into the area prior to wounding is what yielded the results we saw on the previous slide. So for this reason, we moved to a lineage tracing model. This lineage tracing model is when a cell expresses adiponectin, so adipocytes, they are permanently turned green, and all other cells are red. And so if we see a green fibroblast in the wound, we know that it was of an adipocyte lineage. So the first question we wanted to answer is, are these even fibroblasts that we're looking at? And so in this first image, we have some immunochemistry. And you can see in the subcutaneous layer in the bottom part that we do have some green cells. We would expect that because they're adipocytes. But we also have in the dermis some GFP-positive cells that co-express COL1 and alpha SMA, which are both fibroblast markers. We then wanted to look at individual cells. And so we have some FACS plots here. And you can see over time, we have more and more GFP-positive fibroblasts in the wound environment up until day 14, where we have about 10% of all the fibroblasts in the wound are of an adipocyte lineage. And keep in mind, when we are FACS sorting, the protocol is such that we are getting rid of the supernatant, so any adipocytes involved there wouldn't be sorted. We then wanted to look at fibroblast heterogeneity that are derived from adipocytes. And so there are three sort of conserved fibroblast subtypes that we talk about: lipofibroblast, papillary fibroblast, and reticular fibroblasts. We see here that the majority of the adipocyte-derived fibroblasts are of a lipofibroblast lineage. We also have some papillary, about 20%, and then very few reticular. So the sort of dominating subtype here is a lipofibroblast that are derived from adipocytes. The final thing we wanted to do here was think about what is causing these adipocytes, what is telling these adipocytes that they want to be transitioning to fibroblasts to be involved in the wound healing process. And so something that our group focuses on a lot, as you can tell from today, is the mechanical stress. So in collaboration with our friends at the Gertner Lab, we came up with a 3D collagen model where we can actually inject adipocytes into these collagen matrices and then stretch them, thereby giving mechanical input to these cells. And so for this, we have three conditions. We have a strain condition, we have a non-strain condition, and then we have a condition where we use an FAK inhibitor to sort of modulate the amount of mechanosensory input that these cells are receiving. In the non-strain condition, you can see that we have very little COL1 expression and a lot of adiponectin expression. So we're starting with the adipocytes, and here we think we're ending with the adipocytes. So not really any change. However, in the strain condition, we have a lot more COL1 expression and a lot less adiponectin expression. So on the basis of mechanosensory input, we believe we're seeing an adipocyte to fibroblast transition. And then in the strained condition with an FAK inhibitor, we actually see a lot less COL1 and a lot more adiponectin, sort of similar to the first condition. So this convinced us that the mechanical strain might be a pretty important input for these adipocytes to know that they should be transitioning to fibroblasts in the wound environment. So just to conclude here, what we've seen here is that adipocytes do participate in wound healing by, at least in part, transitioning to fibroblasts. We believe that the adipocytes are appearing gradually, so sort of later in the wound healing process, with most of them showing up in this post-operative day 14 time point. We believe that there's heterogeneity among these adipocyte-derived fibroblasts, and that the majority of them are of a lipofibroblast subtype. And we believe that a primary cue for this transdifferentiation is mechanical stress. So some things that we want to do next, we want to ablate the entire subcutaneous layer and look at what happens sort of en masse with these adipocytes and how are they contributing to wound healing as a whole. We also want to look at some inhibitors of mechanical strain in vivo, so doing some small molecule inhibitors, and also some transgenic knockouts. I want to thank my colleagues and my collaborators in the Longacre Laboratory, and my supervisor, Dr. Longacre, and the Holman Committee for the opportunity to speak today. Thank you.
Thank you for that talk. My first question is, why the adipocyte? You know, when I think about adipocytes, I think of them as just a bag full of lipids and kind of not doing much else. Yeah. It seems like a kind of a long road to go from that phenotype towards a fibroblast. Why not something else, another cell type? And what does the cell do with all that lipid that's normally taking up 95% of the volume of the cell? What's happening?
That's a great question. Thank you. So a few things on that. I think proximity to the wound is a really important component to consider there. And the lipid layer is very connected. And so when you have things like mechanical strain, they're all being pulled at once. So it's a really a strong cue for these cells that they should be doing something. So I think that that's sort of our hypothesis, that local area, there's a lot of signaling, and just by proxy of just literally their approximation to that site of injury, that signaling is allowing them to get the things that they need to transition to another cell type. And I think really importantly, there's been a lot of studies on the plasticity of adipocytes sort of all over the body. And it's not just adipocyte to fibroblast transition that other literature has shown. Of course, in wound healing, that's probably the most important transition, but that's an important thing to consider with this cell type. As in reference to your sort of the lipid-laden components of adipocytes, I think that's partially why we're seeing mostly lipofibroblasts. So of course, they're going to have a more of a lipid-laden cell type. I'm not sure exactly what. There's actually a really great paper by Dr. Horsley that talks about the lipolysis that is sort of the pinning from the transdifferentiation component and how that the lipid is being broken down as it transitions from an adipocyte to a fibroblast. But I think it's important to think about what exactly is happening with that and why. It's a great question.
Great job. Thank you very much. I'm struck by this idea that there are environmental cues in the mechanical stress that occurs, and it's so relevant to everything that we do as surgeons, from open surgery to laparoscopic surgery, wound protectors, to putting those big retractors in and pulling and leaving things for 6 hours while we do our operations. And maybe this is to Dr. Leeper's question, which is, you have all this fat, and you keep talking about the environmental cues, but they can't all be proteins. There are chemoattractants in that wound that are changing the phenotype of cells beyond adipocytes to fibroblasts. So why can't you use metabolomics? I mean, we do, and we get metabolites below 5,000 kilodaltons, in size 2, 3, 1,000, and it's beyond these sort of complex proteins that you think are the only things that are signaling peptides to the next cell line downstream. I think that's important.
Yeah. Oh, yeah. Absolutely.
So can you talk about what's in the mechanical stress that's starting this entire process? What's the most proximate event?
Yeah, I think that's a really, really great question, and something that we would love to study further. I think that there is a lot of challenge in sort of, as you mentioned earlier, with the whole secret to, like, being able to actually measure things that are non-protein, non-DNA based events in that area is really challenging. We'd love to work further with that. I think we're still kind of thinking about how can we look at these cells really individually and think about what is actually happening to them exactly as you're describing. I hope that you'll see more from us in that in the future, but I don't know a great answer to that right now. Yeah, thank you.
And then just one quick question from the audience. Aside from their cell surface markers, what other evidence is there that they're actually functionally acting like fibroblasts?
Sure. So again, this project right now, I think, is in its infancy. So we're looking at the entire matrix of these cells and what they're producing from a single cell level right now. And so I think I'll have more data on that for you at a later point, but we're hoping to show that they are not only, of course, looking like fibroblasts, but also acting like fibroblasts. Yeah.
Great. Thank you very much.
Thank you.
Thank you so much for the opportunity to speak today. I'm really excited to share with you some of our latest results. As was alluded to earlier, this work was recently published, and we're kind of now in the next steps of translation. So in 2015, we reported that the cellular culprit for skin scarring, at least on the dorsal skin, is an embryonic lineage of fibroblasts, the engrailed 1 lineage. So these engrailed 1 positive fibroblasts, or EPFs, emerge in the dorsal skin of developing mice shortly before a transition from scarless healing, which characterizes the embryonic period, this fully regenerative healing, to scarring healing, which prepares the animal for life outside the womb. So if we take a step back and think about the origins of the engrailed 1 lineage, what we thought in 2015 was this is purely an embryonically derived lineage. So engrailed 1 lineage negative fibroblasts at some point during gestation activate this Hox gene, they become engrailed positive fibroblasts, and these embryonically derived EPFs contribute to scarring in postnatal life. But there was another possibility about 3 or 4 years at this point that we had considered. What if engrailed 1 lineage negative fibroblasts in postnatal life activate engrailed, making these postnatally derived EPFs that contribute to scarring? And this may seem kind of like a semantic thing, but actually this distinction matters, because if engrailed can activate postnatally, it means that we can interfere with postnatal ENF to EPF transition and potentially yield ENF-mediated healing, or engrailed 1 lineage negative fibroblast-mediated healing. And I'll remind you that engrailed negative fibroblasts are the predominant type fibroblasts during the embryonic period where scarless healing occurs. So we wondered, would ENF-mediated healing also be regenerative? So the first step is just see what ENFs and EPFs do in wounds. So we fractionated by flow cytometry ENFs and EPFs in red and green respectively, and then transplanted them into the dorsal skin of Black 6 recipient mice. And then we wounded in the area where we engrafted, and we looked by fluorescence histology to see what the cells are doing. So if you look in the top row, you'll see in unwounded skin, EPFs and ENFs both just maintain this quiescent morphology. They're not really actively laying down collagen, as we would expect. And in scars, engrailed positive fibroblasts, as you see in the bottom left panel, take on this activated morphology, this stellate cell shape. They begin secreting collagen. They're doing the things that we expect a scar culprit fibroblast to be doing. What we were really surprised to see, though, was that in ENF engrafted scars, as you see in the bottom right, there were lots of GFP positive cells around. And actually, we could see some fibroblasts that were, I guess you could say, orange, or in the middle of reported recombination, indicating that the fibroblasts we had engrafted had either begun activating engrailed, or the other possibility is we just inadvertently had transplanted a couple of GFP positive cells, and that's what we're seeing. So to confirm that postnatal engrailed 1 activation truly occurs, we developed engrailed 1 Cree ERT mice. So here we give tamoxifen and then we wound. So GFP positive cells in the scar necessarily represent fibroblasts that activated engrailed. And as you can see in the bottom row, about 40% of the scar fibroblasts are GFP positive. So about 40% of the scarring EPF lineage is actually postnatally derived. So then comes the question, what explains engrailed activation in postnatal life? And as you've heard several talks today talk about, mechanical forces are really fundamental to the scarring response. And I won't belabor the details of it, except to say that in the FACT pathway, which you've heard about earlier today, the final transcriptional effector is a protein called YAP, or yes-associated protein. And YAP translocates into the nucleus, where it binds DNA and activates pro-proliferation and migration genes. If we actually found just off the shelf a previously FDA-approved drug called verteporfin, which inhibits YAP's function, which we used in these experiments that you see here. So what we did in this experiment was produce a dorsal linear incision, again in the backs of these engrailed Cree ERT transgenic mice, and then applied a mechanical erector set over the healing wound to apply mechanical force across it. And the result of that, as you see in the top panel in the third image over, is this thick hypertrophic scar. But if we pre-treat the wound with verteporfin, this inhibitor of yes-associated protein, you find that the scar gross appearance is dramatically improved. And when we look at the fluorescence histology, we see that the level of engrailed 1 activation, as denoted here in the GFP positive cells, is dramatically lower. So this was our first indication to us that blocking mechanotransduction signaling can block the activation of engrailed in a wound context. So is the result wound regeneration? Here you see, rather than an incisional model, this is an excisional model. So we produce a circular incision and then stent it open. And as you can see, in dramatic contrast in the middle row to our PBS saline control, where we end up with a scar bare area, we're really surprised to see that verteporfin-treated wounds were sprouting thick tufts of hair by post-op day 30, as you see in the bottom middle. And by 90 days, the wound area was grossly indistinguishable from unwounded skin, suggesting that we had achieved wound regeneration. Looking by the histology, we see that by 30 days, as demarcated with the white arrows, there are several structures that look like hair follicles and glands emerging. And by 90 days, again, our verteporfin-treated wounds were histologically indistinguishable from unwounded skin. And these structures stain for cytokeratins 14 and 19, oil red. So it indicates that we've achieved regeneration of hair follicles and glands within what would otherwise just be a bare scar area. And finally, looking at the lineage tracing, our control wounds are predominantly healed by EPFs, whereas our verteporfin-treated wounds are predominantly healed by ENFs, as shown here in red. We developed a machine learning algorithm as well to quantitatively compare histological specimens by their matrix ultrastructures. And again, I won't belabor the details, but the net of this was that at a quantitative level, the matrix ultrastructure of verteporfin-treated wounds was also indistinguishable from unwounded skin. And as a result of having the same ultrastructure of unwounded skin, our verteporfin-treated wounds had the same mechanical strength as unwounded skin. Scars, of course, being typically weaker than unwounded skin. Now, with any chemical inhibitor, there's always the potential for off-target effects, and so we've also generated YAP knockout transgenic models mimicking our verteporfin treatment strategy. Here, it's limited again to the engrailed lineage, and you can see both grossly in the middle and bottom rows, as well as histologically, we see the same wound regeneration at 30 days with recovery of normal hair follicles and glands. And finally, we've developed transgenics to directly go after the postnatal engrailed one activating fibroblasts. So here, it's a diphtheria toxin receptor model where we wound and then give DT 3 times to ablate all fibroblasts expressing engrailed. And finally, just not to beat a dead horse, but it's the exact same result. Grossly, we see regeneration of hair follicles. By H&E and by our IF, we see the same recovery of hair follicles and glands and normal appearing skin. So in conclusion, we've shown that postnatal engrailed 1 negative fibroblasts activate engrailed through a YAP-mediated mechanotransduction signaling mechanism. And we've shown that engrailed 1 activation occurs in the wound environment as a result of mechanical force sensing. And finally, we've shown that if you inhibit YAP mechanotransduction signaling, we can achieve wound regeneration with recovery of normal hair follicles, glands, matrix ultrastructure, and mechanical strength. And so we're really excited by these results, and we're now taking this work into pig to see what the efficacy of verteporfin treatment is in preventing scarring in that model, it being closer to human skin. I'd like to thank my mentor, Dr. Leinker, as well as another MSTP student, a colleague of mine, Heather Desjardins-Park, Michael Januszyk, who you've heard from earlier today, as well as Michael Davitt, a postdoc in the lab, all of whom have been really instrumental to this work. And I'm happy to take any questions you may have. Thank you.
Thank you. I guess one obvious question based on Dr. Guardino's talk right before you is, how do you kind of contextualize what's going on with adipocytes in response to mechanical forces and what's going on with fibroblasts and mechanical forces? And since you're able to completely get regeneration by blocking the fibroblasts, does that imply that the adipocyte mechanotransduction and whatnot is not very important? Or how do you contextualize all of that?
Yeah, so I actually going to go back to this slide. So one of the features that we tend to see with our verteporfin-treated wounds is actually a certain degree of fat hypertrophy. So if you look at 14 days, the verteporfin-treated wounds are a bit fattier than their PBS saline counterparts. I think there's this sort of this broad theme that I guess multiple talks today have touched on. So I think wound healing, or I guess fibroblast contributions to wound healing, is extremely plastic, and I think it's sort of a whoever is around contributes. So we've done some experiments where we've taken fibroblasts fractionated from different anatomical layers, so the papillary layer fibroblasts, deeper down reticular layer fibroblasts, even deeper down hypodermal fibroblasts, put them into stiff environments just to see what do they do when they're presented with those the contractile forces, the mechanotransduction signaling. And it's interesting because the end result may sometimes be different, but the pathways there are not always the same. So if you look at the papillary layer fibroblasts, it's interesting because they're not particularly mechanoresponsive at all, but the deeper down layer, reticular layer fibroblasts are extremely mechanoresponsive. We see large-scale upregulation of the fibrogenic program. We actually see activation of engrailed. We can catch it by bulk RNA sequencing, which is engrailed is sort of on very quickly, so it's actually quite hard to catch by transcriptomics. And then for the deeper down hypodermal fibroblasts, we find that they kind of go down one of two paths, either continuing their lipogenic program or this massive upregulation of collagen. So, and then we also see certain mechanotransduction signaling pathways upregulated in that context. So, I think there's kind of there's sort of this issue that these are all many different lenses that we're choosing to look at fibroblast heterogeneity, and at the end of the day, they will line up in some ways and not in others, and the fundamental mechanisms may or may not be the same. So it's very messy, very complicated.
Right. Yeah. So what's the role of, or this is from Slido, or what do you know of ENF to EPF transition in pigs and humans?
So in pigs, I should say that this is very much trial work that's happening right now. We've had one, I guess you could say, trial pig, and then we have 2 currently going now. It's difficult to say. So I can say from a gross perspective, yes, the wounds grossly look less fibrotic, and we've tried the same machine learning approach, showing similar matrix ultrastructure to unwounded skin. We've done some mechanical strength testing, but we can't lineage trace in pigs, which obviously one of the benefits of starting this work in mouse is that we can understand the fibroblast lineages better. With regards to what's happening in pig, we have to rely then on our surrogate markers for lineage, like CD26. So we haven't done that IF yet, but certainly that would be very important to look at.
But do pigs and humans have ENF and EPF fibroblasts? I think that's the question.
Yeah, I guess the pig and human counterpart to the ingrailed negative and positive lineages needs to be worked out for sure. I should say that there's some as a result of having to use CD26, I believe there's been some work looking at CD26 positive fibroblasts and correlating them to the ingrailed positive lineage, but I don't know necessarily what we would correlate to the ingrailed negative lineage, so it's hard to say negative to positive how that's occurring in humans, or if it does at all.
Okay. Would you like?
Great presentation. Thank you very much. That verteporfin, that's a great name. It's got good onomatopoeia to it. But I wondered, you sort of glossed over the dose response curves that you must have generated to get to this level of efficacy. And then I wondered, if you've seen any toxicity, because usually with these, you hit the sweet spot of the dose, you get the response you want.
Right.
And then when you go over, you get actually a toxic response.
Yeah, absolutely.
Is that possibility there?
Yeah, so absolutely. And part of the reason we generated our transgenic models was exactly because reviewers said, well, what about your possible off-target effects and toxicity? So 100%, that's absolutely correct. There is a dose responsiveness. This was one of the supplemental figures for this paper, which just for time reasons I didn't add here. But yeah, if you start going beyond your therapeutic dose, absolutely, there's a reduction in the regenerative phenotype. So you have this sort of, I guess you could say just a bell-shaped curve, basically, our dose responsiveness. And so for us, we typically use 1 mg per mil and then inject 30 microliters of that concentration. Going up, we've done 1, 2, 4, 8. By the time you get to 4 and 8 mg per mil, the regenerative phenotype is very much, you start getting into delays in wound healing and various problems as a result of off-target effects.
And then I think we have time for one final question from Slido. Thinking about human applications and other areas of wound healing, could this have implications for the treatment of other fibrotic processes?
Yeah, so YAP inhibition seems to be useful for organ fibrosis broadly. There were 2 papers actually published within the last 2 months. One using YAP inhibition for liver fibrosis and one YAP inhibition for kidney fibrosis. And in both contexts, it looks like it has efficacy there. This is obviously in skin fibrosis, but it remains to be seen how globally involved YAP is in mechanotransduction and fibrotic mechanisms throughout the body. I will say, at least as far as the ingrailed mechanism, as Dr. Foster mentioned earlier, that is something that we see restricted to skin, but that doesn't mean that there aren't corollary populations elsewhere. We just don't know yet.
Okay, thank you.
Yeah, thank you.
Good afternoon. My name is Siddharth Menon. I'm a third-year PhD student in Dr. Longacre's lab, and thank you for the opportunity today to share my research with you. The calvarial sutures are a fibrous mesenchymal tissue that form the margins between the bone plates of the calvarium. They're highly conserved among species and serve as the major site of postnatal calvarial morphogenesis and develop in close coordination with the brain. Craniosynostosis is the pathological premature fusion of one or more cranial sutures and presents with significant clinical problems such as exothalamus, increased intracranial pressure, midface hypoplasia, to name a few. The mouse and human calvarial sutures are highly conserved. The PF suture, metopic in humans, is the only suture that fuses early postnatally, while the remaining sutures remain patent into late adulthood. The similarities between the mouse and the human calvarium provide a suitable model for studying calvarial suture biology. What we know from previous work in our lab is that the mouse PF suture fuses by postnatal day 15 through a process of endochondral ossification, while the SAG and the coronal, not shown here, remain patent into late adulthood. Active canonical Wnt signaling in the suture mesenchyme maintains suture patency, while inhibition can drive fusion. And finally, the suture mesenchyme is a niche for skeletal stem cells and osteoprogenitors that work in coordination in this microenvironment with the overlining periosteum, the underlining dura mater, and the osteogenic bone fronts to coordinate cranial suture morphology and calvarial development. The identification and specification of the mouse skeletal stem cell by our group in 2015 fueled our investigation into whether the mouse mouse SSCs are resident in the calvarial suture and if they play a role in physiologic and/or pathologic suture fusion. We started our investigation with time course profiling of SSCs from the PF, SAG, and coronal sutures in mouse along the timeline of PF suture fusion, which is postnatal day 3 through 17. And what we found is that in the PF suture, the only suture that fuses physiologically, that there was a decrease in representation of SSCs in advance of fusion, whereas in the patent SAG and coronal sutures, this representation stayed more sustained through this time course. We next turned our attention to models of pathological suture fusion or craniosynostosis. And Sethray-Chotzen syndrome is characterized by unilateral or bilateral coronal craniosynostosis as a result of loss-of-function mutations in the TWIST1 gene. And the TWIST1 haploinsufficient mouse is a suitable model for studying this form of coronal craniosynostosis, faithfully recapitulating the human condition. And in these TWIST1 mice relative to their wild-type coronal sutures, we see that there is a decrease in the representation of skeletal stem cells in these pathologically fusing sutures versus wild-type controls. Given these observations, we then turned our attention to a series of experiments that were previously performed in the lab by a postdoc, Dr. Ben Bjorn. In these experiments, Bjorn was able to demonstrate that activation and/or inhibition of canonical Wnt signaling could alter the suture fate, either driving patency or inducing fusion. We wanted to understand how canonical Wnt signaling might be manipulating SSC frequency within these sutures. And what we found is that in the context of activation of canonical Wnt signaling in the suture, there was an enrichment of this SSCs within the patent suture. But in the context of Wnt inhibition, we saw a decrease of the skeletal stem cell representation and eventual suture fusion. So what have we seen so far? We've learned that SSC frequency decreases in advance of both physiologic and pathologic suture fusion. However, activation of canonical Wnt signaling increases SSC's representation, and this can result in suture patency. So we asked the question then, can enhanced activation of canonical Wnt signaling potentially rescue a craniosynostotic phenotype? To explore this question, we developed a twist axon compound mutant with the knowledge that the TWIST1 mouse harbors a coronal craniosynostosis, while the axon 2 mouse is in a model of enhanced activation of canonical Wnt signaling. And we were curious about the frequency of coronal craniosynostosis in this compound mutant, twist axon compound mutant relative to twist mice alone. To our delight, we actually found that the coronal sutures from this compound mutant had a largely undifferentiated mesenchyme in compared to the twist coronal sutures, more similar to wild type. And upon FACS sorting, we see that there's a restoration of the representation of skeletal stem cells to near wild type levels relative to the twist mouse alone. And this correlated to a complete suppression of bilateral or unilateral coronal craniosynostosis in the compound mutant relative to the TWIST1 mouse alone. We screened 250 animals from postnatal day 6 through 6 months in age. And I hope you can see here that the compound mutant remains patent out to 1 year in age and by micro-CT. And then we can see that there is active canonical Wnt signaling in the patent suture mesenchyme even out to 6 months in age. The blue staining, exgal staining, is a readout of active canonical Wnt signaling in this mouse. Again, this suggests the importance of canonical Wnt signaling to suture patency. So we next asked the question, can we use this knowledge for some level of translational intervention? The conventional treatment for craniosynostosis is a suturectomy, and quite often there is a subsequent resynostosis requiring follow-up treatment. And we wanted to understand if transplantations of skeletal stem cells could either prevent or delay the resynostosis and prevent a subsequent retreatment. So we developed a surgical procedure in which postnatal day 16 twist-1 mice coronal sutures were excised and replaced with a collagen sponge seeded with SSCs and recombinant Wnt3a protein. And we monitor the extent of refusion by micro-CT and histology over a 14-week period. After 14 weeks postoperative, we found that SSCs alone, our sponge alone, no treatment, and the Wnt3a groups refused or nearly refused respectively over this period. However, those treated with SSCs and Wnt3a remained relatively patent during this time course, suggesting a synergistic relationship between the Wnt3a and SSCs transplanted into this suturectomy site. To assess the role of these transplanted SSCs to prevent resinostosis, we use GFP-positive SSCs in a combination with Wnt3a, and I hope you can appreciate the contribution of these GFP-positive transplanted cells in developing the newly engineered suture mesenchyme, and in addition to recruiting endogenous cells into this mesenchyme. In contrast, SSCs transplanted alone contributed to the refusion and synostosis of the suturectomy site. So in conclusion, proper balance of skeletal stem cells is crucial to determining suture patency or fusion, and canonical Wnt signaling is critical to maintaining this balance. Engineering a suture mesenchyme by transplantation of skeletal stem cells combined with Wnt3a protein can prevent resinostosis post-suturectomy. And we actually view skeletal stem cells as a potential tool suitable for preventing bone formation rather than simply engineering new bone. With that, I'd like to thank Dr. Longacre for the opportunity to work on this project in his lab, Dr. Natalina Corto, my mentor in the lab, and Dr. Chan and Dr. Wan for their support on this project and my PhD throughout the years, the entire Longacre lab, and my funding resources. Thank you.
Thank you for that really great talk.
Thank you.
I was trying to think about translation and wondering if, Wnt signaling is important throughout the body. Is this process specific to these sutures in the skull, or does this also affect the long bone fusion during development? And so, if so, how will you target this bone growth versus others?
Right. So, yeah, Wnt is critical in most regenerative processes, especially in the context of injury. This is a proof of concept model in which we wanted to show that really to prevent the resinostosis, you need to have the right niche cues and environment. And so our Wnt treatment is not targeted specifically to the SSCs because it can also have effect on endogenous cells. I would expect that it has a different effect on the osteogenic fronts, which are more differentiated. We know that Wnt can have a dual effect on upstream skeletal stem cells versus more fate-committed cells. It can have alternate effects. So in reality, it would require some fine-tuning of the experimental procedure and more targeted activation of Wnt in skeletal stem cells particularly.
I would just say great presentation, and I really enjoy everybody's blending of molecular science with great imaging, because we're all visual animals and we need to see sort of what the end product is. Is there a mouse model for Wnt3a overexpression or forced expression? And what might you predict would happen? And how does that play out in patients in vivo, in your mind? Do patients sometimes make too much Wnt3a?
Yeah, that's a good question. There are, as far as I know, there isn't a mouse model of Wnt3a specifically, but there are GSK models of inhibitors and other aspects. And in the context of the clinic, that's a good question. It's unclear what might be causing or what the Wnt3a is doing to these skeletal stem cells. There could be a series of downstream effectors that manifest in the clinical situation that is not something we would see from Wnt3a specific.
I'd like to thank the department for the opportunity to speak today. I'm going to be talking about kind of the final little piece of the project I've been working on for the past 3 years, looking at the skeletal stem cell response after tendon to bone injury. Thankfully, Sid and Lauren have already spoken today to talk to you about the MSSC hierarchy. The MSSC differentiates into BCSPs, which then in turn become cartilage, bone, and stroma. We isolate the MSSC through FACS sorting, flow cytometry, as demonstrated in the figure next to it. I've previously shown during Holman Day that MSSCs are in elevated levels within the TBI following injury, suggesting that maybe the MSSC actually plays a role in not just bone formation and bone morphogenesis and remodeling, but maybe as well in tendon to bone healing. We found that there was a transcriptomic difference in injured MSSCs compared to control MSSCs. You can see this very clearly by seeing the injured group in light pink and the uninjured group in the light blue. And then specifically, we saw a downregulation in the TGF-beta signaling cascade in the injured MSSCs isolated from the injured TBI. So we wondered if functional modulation of TGF-beta alters the MSSC in response to TBI healing. We utilized a model that we have previously discussed where we do a partial tenotomy at the tendon to bone interface, and we used wild-type mice as well as a transgenic that we developed, which I'll talk about in a little bit, and we utilized immunofluorescence, gross evaluation, FACS sorting, and confocal microscopy. First, we wanted to confirm our findings from RNA-seq, specifically that there was a downregulation in TGF-beta signaling. So we looked at the protein level. Specifically, we looked at the pSMAD2/3 expression, which is a protein that is phosphorylated by TGF-beta and then induces TGF-beta process. And very excitingly, as you can see here in the light green is the pSMAD expression. The control group has a bright expression with over 40% of the cells having nuclear expression, but this is significantly decreased in the injured group. The picture to the left of that, or to the right of that, is just the nuclear staining, and then we have the combined images. So overall, this suggests that there's not only a decrease in TGF-beta signaling at the RNA level, but also the protein level. So we wondered if, in the exogenous model, if there would be an influence on the quality of healing as well as in the MSSC response. First, here we can see the saline control where there is gross healing by day 7. There's a little bit of inflammation and a little bit of fibrosis. Here, with the use of exogenous TGF-beta, healing is significantly delayed, where there is still not complete healing by day 14 and a lot of fibrosis. Here we used SB, which is a TGF-beta 1 inhibitor, and there was improved healing by day 7 and minimal inflammation and scarring. We then looked to see if there was a response with exogenous TGF-beta with MSSCs. Here we see that there is the same increase in MSSCs from day 3 to day 7 that was a significant increase. This is blunted when there's use of exogenous TGF-beta, but then when we use SB, the TGF-beta inhibitor, there is a magnification, further expounding the response of MSSCs, as well as BCSPs, the downstream progenitor. We then wondered if the same was true at the endogenous level. We specifically looked at fibrilin mice, which are marfanoid-like, where the fibrilin protein is altered and results in aberrant expression of TGF-beta. In looking at our control mice, the DMSO-treated mice, there is poor wound healing, poor tendon to bone healing, and there is not complete healing by day 14, and a lot of inflammation. However, this is rescued when we use SB, where there is pretty much complete healing by day 7 and minimal inflammation and fibrosis. Looking at our endogenously elevated TGF-beta levels, there is no MSSC increase after injury. However, this response is rescued with the use of SB, both in MSSCs as well as BCSPs. So in conclusion, we have identified that MSSCs increase in frequency following TBI injury. This response is dampened by TGF-beta, while TGF-beta inhibition reduces or rescues MSSC response and allows for improved healing, and may possibly help with regeneration in the future. I'd like to thank Dr. Longacre, Dr. Quarto, and everyone in the Hagee Lab, as well as the ACS and TTE for funding of this project. Thank you for your time.
Thanks. Very nice presentation. One thing that I was, as your data was going by, it seems like the MSSCs, they only go up several fold. Is that surprising to you, or is that expected based on how bones and tendons and heal?
I was surprised, even in the beginning, when we actually saw an increase in MSSCs after injury. So that in and of itself was a notable finding for us, and that it completely goes away with TGF-beta.
But wouldn't you think that, like in the context of an injury, that those cells would go up? And why was it a surprise then?
It was a surprise because in the data not shown here, when we did an injury in the mid-substance, there was no change in MSSC level, but only when it was close to the bone. So there's something happening even though the tendon's being injured.
And how does this relate to how the tendon-bone interface is formed developmentally? I mean, are the same factors involved, or is it a different process?
Right now, we don't actually know if MSSCs do play a role in tendon-to-bone healing. That is something I would definitely like to look at in the future, so I don't have an answer for that yet, but hopefully one day.
Great job. Thank you very much. I really just appreciate the, again, blending of molecular science with the visual and functional output in this case. I have a similar question to the one I had before, which is something about the dose-response curve for you to get to the dose of TGF-beta that gave you the sort of perfect response. And, maybe this is to what Dr. Gertner's getting at, which is how do we understand how to dose and intervene in a healing process in a perfectly timed and perfectly efficacious manner. And so I would ask you again, as I asked before, is how did you arrive at this dose? And then is there a toxicity if you go higher?
So I used previous research to find the dosing that I used for the SV molecule. Haven't yet seen the toxicity, but I'm sure it does exist if I went to higher doses, and something I plan on definitely doing in the future.
And just one final clinical question. What's the failure rate of clinically in the bone to tendon kind of insertion, which is kind of, I think, what you're trying to recreate or strengthen? Is that fail a lot clinically?
From the fail after I do the injury, or what do you mean exactly?
No, after you put in a bone anchor and drill some holes and put a screw in, and is that like a high failure rate area for orthopedic surgery or hand surgery, or is it? It's just a question of what is the failure rate? Is it 1%? Is it 20%?
So what I was trying to recreate was sort of the not spontaneous rupture, because I wasn't doing a complete injury, and not after necessarily after a surgery. It was more just if there was a partial injury.
I see.
So I don't know the answer to that.
Oh, okay, great. Okay, thank you. I think that's the end of the session. We will adjourn until 3 o'clock and then come back for the clinical papers. And thank you, everyone, for your attention.